and biotechnology. Genetic engineering deals with the modification of genetic material of an organism. It is the process of using recombinant DNA or rDNA technology to alter the genetic makeup of an organism. Recombinant DNA is a technology that uses enzymes to cut and paste together DNA sequences of interest. The recombined DNA sequences can be placed into vehicles called vectors that ferry the DNA into a suitable host cell where it can be copied or expressed. So, recombinant DNA technology is the joining together of DNA molecules from different organisms and inserting it to an host organism to produce a new genetic combinations that are of value to science, medicine, agriculture, and industry. Genetic engineering, it involves manipulating genes in order to make useful products. Vector, it is used to carry a desired DNA sequence into a host cell. Plasmid, it is a small cellular DNA found in bacteria. DNA ligase, its function is to stick cut pieces of DNA together. Restriction enzyme, it is used to cut the plasmid at specific sites. Recombinant DNA technology is a technique in which a fragment of DNA from another cell or organism is isolated and then inserted into the DNA of another cell or organism. This allows scientists to introduce a new characteristic into an organism by inserting a new gene into it. Recombinant DNA technology involves four major steps. Firstly, a DNA fragment containing the gene of interest is obtained from a donor cell. Secondly, a suitable plasmid is obtained from a bacterium. Plasmids are commonly used as vectors to transfer the gene of interest into a host cell for expression. Thirdly, the DNA fragment containing the gene of interest to cut using an enzyme called restriction enzyme. This enzyme acts like scissors. It recognizes specific base and cut the DNA at a specific point. On the other hand, the same restriction enzyme is used to cut open the plasmid. Lastly, the DNA fragment containing the gene of interest is inserted into the open plasmid with the help of another enzyme called DNA ligase. This enzyme acts like glue. It catalyzes the joining of the DNA fragment and the plasmid. And then a recombinant plasmid is firmly formed. This recombinant plasmid can be then introduced into a host cell for different purposes, and such as producing proteins of other species like human insulin or producing genetically modified organisms that possesses new characteristics. How genetic engineering can help humans. With the advent of genetic engineering, scientists can now change the way genomes are constructed to terminate certain diseases that occur as a result of genetic mutation. In medicine, genetic engineering has been used to mass produce insulin, human growth hormones, follicism for treating infertility, vaccines, and many other drugs. In research, organisms are genetically engineered to discover the function of certain genes. Genetic modification of an animal involve altering its genetic material by adding, changing, or removing certain DNA sequences in a way that does not occur naturally. It aims to modify specific characteristics of an animal or introduce a new trait such as disease resistance or enhance growth. Example, inserting growth hormone to make small animals grow bigger than usually they are. Application of genetic engineering and research on plant gene. Genetically modified plants have been engineered for scientific research to create new colors in plants, deliver vaccines, and to create enhanced crops. Genetic engineering on plants are ways to protect plants from damaging pests and diseases. To further understand genetic engineering on plant gene, watch this video.
Tomato. Doesn't this term sound a little weird? You might be wondering, is it a tomato or a potato plant? It's actually both. Yes, you heard me right. In this single plant, the tomatoes grow above the ground while the potatoes grow below the ground level. And how is this made possible? It can be done by a process called grafting. In this, the stems of both the tomato and the potato plants are chosen and then joined together by tying them. This is an example of plant engineering. On the similar lines, when we cut and join genetic material, the process is known as genetic engineering. It's